Welcome to this scientific lecture on how artificial intelligence and machine learning are revolutionizing our understanding of the origin of life. Over the next 16 minutes, we'll explore the cutting-edge computational methods that scientists are using to tackle one of humanity's most profound questions. The origin of life occurred approximately 4 billion years ago, when simple chemical compounds somehow self-organized into the first living cells. This transition from chemistry to biology remains one of science's greatest mysteries. The fundamental challenge in origin of life research is the sheer enormity of chemical space. Let me explain what this means. Chemical space refers to all possible molecules that could theoretically exist. For small organic molecules relevant to life, this space contains approximately 10 to the 60th power different structures. That's more molecules than atoms in the observable universe. Traditional laboratory experiments can only test a tiny fraction of this chemical space. If a chemist synthesized one new molecule every second, it would take longer than the age of the universe to explore even a billionth of the possibilities. This is precisely where artificial intelligence and machine learning become not just useful, but absolutely essential. These computational methods can rapidly explore vast regions of chemical space that would be impossible to investigate experimentally. Let's examine the first major application, using machine learning to predict how molecules will react with each other under prebiotic conditions. When two molecules encounter each other, will they react? What products will form? How quickly will the reaction proceed? These questions are fundamental to understanding how complex molecules could have emerged from simpler precursors. Scientists train neural networks on vast databases of known chemical reactions. These networks learn patterns that govern molecular reactivity. A neural network consists of layers of interconnected nodes. The input layer receives molecular structures encoded as numerical features. Hidden layers extract patterns and relationships. The output layer predicts reaction outcomes. These networks are trained on databases containing millions of documented chemical reactions. The Reaxys database alone contains over 50 million reactions. Training set size: 5 times 10 to the 7th reactions. The remarkable aspect is that the network learns fundamental chemical rules without being explicitly programmed with them. It discovers patterns like electron transfer, bond formation energies, and steric hindrance from the data Let's examine a concrete example: the Formose reaction network. This is a complex autocatalytic reaction that produces sugars from formaldehyde. Formaldehyde, a simple one-carbon molecule, can react to form 2-carbon and 3-carbon compounds. These intermediates then combine to form sugars. The reaction network contains hundreds of possible pathways. Traditional chemical analysis of this system is intractable. There are too many competing reactions occurring simultaneously to solve the equations analytically. Machine learning approaches can predict which pathways will dominate under specific conditions and identify bottleneck steps that limit product formation. This guides experimentalists to focus on the most promising conditions. The second major application is predicting protein structure, which is crucial for understanding how the first functional proteins could have emerged. A protein is a chain of amino acids that must fold into a precise three-dimensional structure to function. The sequence contains typically 100 to several thousand amino acids. This leads to Levinthal's paradox. If a protein randomly sampled all possible conformations, it would take longer than the age of the universe to find the correct fold. Yet proteins fold in milliseconds to seconds. For a 100 residue protein, there are approximately 10 to the 300th power possible conformations. AlphaFold, developed by DeepMind, represents a revolutionary breakthrough in this field. It predicts protein structure with near experimental accuracy. AlphaFold uses a sophisticated architecture. It takes the amino acid sequence as input, analyzes evolutionary relationships through multiple sequence alignments, uses attention mechanisms to identify which residues interact, and outputs a predicted three-dimensional structure. The system was trained on 170,000 experimentally determined protein structures from the Protein Data Bank. It achieves accuracy comparable to experimental X-ray crystallography for many proteins, a feat that won its creator's recognition as one of the top scientific breakthroughs of the decade. How does this relate to origin of life research? Scientists use AlphaFold to reconstruct ancient ancestral proteins that existed billions of years ago. By analyzing modern proteins from diverse organisms and tracing their evolutionary relationships backward, scientists can infer the sequences of proteins that existed in LUCA, the last universal common ancestor. AlphaFold can then predict the structures of these ancestral proteins, allowing researchers to understand what molecular machinery the earliest life possessed. The third application is perhaps the most exciting, using machine learning to discover autocatalytic chemical networks that could have sparked life. An autocatalytic network is a set of reactions where the products catalyze the formation of other products in the network, creating a self-sustaining cycle. Molecule A helps produce B, B produces C, C produces D, and D helps produce more A. Such networks are prime candidates for primitive metabolism because they can maintain themselves and grow without the complex genetic machinery that modern life uses. The challenge is finding these rare autocatalytic networks in the vast ocean of chemical space. Most random collections of reactions do not form closed, self-sustaining cycles. Scientists are using reinforcement learning, a type of AI that learns through trial and error, to discover these networks. A reinforcement learning agent proposes adding reactions to a network. A chemical simulator evaluates whether the network becomes autocatalytic. The agent receives a reward for finding autocatalytic behavior and learns which types of reactions are promising. The reward function is designed to favor networks that exhibit autocatalytic behavior. All molecules in the network can be produced from a small food set, and the network grows when fed these simple inputs. A fascinating case study involves using AI to explore alternative versions of glycolysis, the central metabolic pathway that breaks down glucose. Modern glycolysis consists of 10 enzymatic steps that convert glucose into pyruvate, producing ATP and reducing power. This pathway is universal across life. The question is, could alternative pathways have existed in early life? Are there other ways to break down glucose that might have been used before evolution optimized the current pathway? The number of possible alternative pathways is greater than 10 to the 15th. Machine learning algorithms have discovered novel pathways with fewer steps, or using different chemical logic that could potentially be more accessible to prebiotic chemistry. The fourth application focuses on the RNA world hypothesis, which proposes that early life was based on RNA molecules that serve both as genetic material and as catalysts. An RNA molecule is a sequence of 4 nucleotides: adenine, cytosine, guanine, and uracil. This sequence folds into a complex three-dimensional structure that determines its function. The challenge is to design RNA sequences that fold into structures capable of catalyzing specific chemical reactions. This is a reverse problem. Given a desired function, find sequences that perform it. Scientists use generative models, particularly variational autoencoders, and generative adversarial networks to design RNA sequences. A variational autoencoder learns to compress RNA sequences into a low-dimensional latent space that captures essential features. By sampling from this latent space and decoding, we generate novel RNA sequences with desired properties. These models are trained on millions of natural RNA sequences and their experimentally determined structures from databases like RFAM and the RNA Structure Database. The output is novel ribozymes, catalytic RNA molecules that do not exist in nature but possess desired catalytic activities. These give us insights into what RNA catalysts might have been possible in the RNA world. Let's do a back-of-the-envelope calculation to understand the scale of the problem and why AI is necessary. Consider a simple prebiotic soup containing just 50 different molecular species. This is a conservative estimate. The actual number could be much higher. The number of possible binary reactions, where 2 molecules react, is 50 choose 2, which equals 1,225. But each pair of reactants could produce multiple different products through different pathways. Let's conservatively estimate 3 possible products per reaction pair. Total reactions approximately equals 1,225 times 3, which is 3,675. Now, the number of possible networks you can form by selecting subsets of these reactions is 2 to the power of 3,675, an incomprehensibly large number. This equals approximately 10 to the power of 1,100, vastly larger than the number of atoms in the observable universe. If we could test one network per microsecond, it would take 10 to the power of 1094 years, far longer than the age of the universe, which is only about 10 to the 10th years. Artificial intelligence reduces this search space by orders of magnitude, by learning chemical principles that eliminate vast swaths of impossible or uninteresting networks, focusing computational resources on promising regions. The fifth application involves using machine learning to accelerate molecular dynamics simulations of prebiotic environments. The early Earth's oceans were chemically complex environments with varying pH, temperature, mineral surfaces, and high ultraviolet radiation. Simulating molecular behavior in these conditions is computationally expensive. Traditional molecular dynamics requires quantum mechanical calculations at every time step to determine how molecules interact. This limits simulations to nanoseconds for small systems. The computational cost scales as order n cubed per step, where n is the number of atoms. Machine learning potentials use neural networks trained on quantum mechanical calculations to predict molecular forces much faster. Neural networks are trained on a database of quantum mechanical calculations. Once trained, the network can predict molecular energies and forces in milliseconds instead of hours, achieving a million-fold speedup. Speedup approximately equals 10 to the 6 times faster. This enables simulations spanning microseconds to milliseconds, allowing scientists to observe rare events like spontaneous vesicle formation or the assembly of protocells. A compelling case study is simulating lipid self-assembly into vesicles, which are proposed as primitive cell membranes. Amphiphilic molecules have a hydrophilic head and a hydrophobic tail. In water, they spontaneously assemble into bilayer vesicles with the tails pointing inward and heads pointing toward the water. Machine learning accelerated simulations reveal the detailed pathways by which these vesicles form, including intermediate structures and the minimum concentration needed for assembly. The sixth application uses machine learning for pattern recognition in metabolic networks to identify core reactions present across all life. By comparing metabolic networks across bacteria, archaea, and eukaryotes, machine learning algorithms identify a universal metabolic core, reactions present in all domains of life. Graph neural networks, which are designed to analyze network structures, identify conserved motifs and pathway modules that likely existed in the earliest life forms. Approximately 400 core reactions out of 5,000 total reactions are universally conserved. These core reactions are prime candidates for protometabolism, the chemical cycles that sustain the earliest living systems before the evolution of complex enzymes. The most powerful approach integrates multiple AI methods into multi-scale models that span from quantum chemistry to cellular behavior. At the bottom, quantum mechanics calculates molecular properties. These feed into molecular dynamic simulations. Those inform reaction network models. Finally, we simulate protocell behavior. AI methods accelerate each level. Each level informs and constrains the levels above and below it, creating a self-consistent picture of how life could emerge from chemistry. Despite these advances, significant challenges remain in applying AI to origin of life research. First, training data is limited. We have extensive data on modern biochemistry, but very little on prebiotic chemistry. Models trained on biological data may not generalize to prebiotic conditions. Second, AI models are best at interpolation within their training distribution. Origin of life requires extrapolation to chemical regimes that may never have been experimentally explored. Third, we lack clear definitions of life and success criteria. What exactly are we searching for? This makes it difficult to design appropriate reward functions for reinforcement learning. Fourth, despite speedups, computational costs remain high for truly comprehensive searches of chemical space. We can explore vastly more than before, but still only a tiny fraction of possibilities. Looking forward, several exciting directions promise to advance the field further. Active learning frameworks where AI algorithms design and guide experiments, creating a closed loop between computation and laboratory work. The AI proposes experiments, robots execute them, and results improve the models. The AI model proposes an experiment. A robotic laboratory executes it. Results update the model, which proposes the next experiment. This closed loop accelerates discovery exponentially. Integration with quantum computing for exact simulation of quantum chemical processes. Quantum computers could simulate molecular systems with exponentially greater accuracy than classical computers. Multimodal learning that combines diverse data types, chemical structures, spectroscopic data, microscopy images, and even geological records of early Earth conditions. Evolutionary algorithms that mimic natural selection to evolve chemical systems in silico. Starting from simple molecules, these algorithms evolve increasingly complex and functional systems. The use of AI in origin of life research raises profound philosophical questions. Can artificial intelligence discover principles of life that human scientists haven't even imagined? The algorithms aren't constrained by our preconceptions about what life must be. This opens the possibility of discovering alternative biochemistries that could exist in environments very different from Earth, relevant to astrobiology and the search for extraterrestrial life. What if AI discovers that multiple independent pathways to life exist? This would suggest life's emergence is more probable than we thought, and that the universe may be teeming with life. Let me highlight some recent breakthroughs that demonstrate the power of these approaches. In 2023, researchers used reinforcement learning to discover a novel autocatalytic peptide network that wasn't known in biology but could function as a primitive metabolic cycle. In 2024, scientists used AlphaFold to reconstruct significant portions of the leucoproteome, providing insights into the molecular machinery of the earliest life. Also in 2024, machine learning identified previously unknown prebiotic synthesis pathways for RNA nucleobases that work under plausible early Earth conditions. Let me summarize the key insights from this lecture. First, chemical space is astronomically large, far too vast for traditional experimental or even computational exploration without intelligent search strategies. Second, AI and machine learning dramatically reduce the effective search space by learning underlying chemical principles that eliminate impossible or uninteresting regions. Third, different AI methods address different scales and aspects of the problem: neural networks for prediction, reinforcement learning for discovery, and generative models for design. Fourth, the most powerful approach integrates AI-driven computation with experimental validation and active learning frameworks. Fifth, we are entering a new era where computational approaches can explore more chemical possibilities in a day than were explored experimentally in the entire history of the field. It's important to place this work in the broader context of science and technology. Artificial intelligence is becoming not just a tool for analyzing data, but for generating scientific hypotheses. The algorithms propose chemical scenarios that human scientists can then investigate. Machine learning potentials and automated tools are democratizing access to complex simulations that previously required supercomputers and specialized expertise. Progress requires deep, interdisciplinary collaboration between chemists, biologists, computer scientists, and mathematicians. No single discipline has all the necessary expertise. Let me conclude with some final thoughts. Artificial intelligence and machine learning are fundamentally transforming how we investigate the origin of life, enabling exploration of chemical space at scales that were previously unimaginable. We are not yet close to solving the problem of life's origin. The chemical space remains vast, and many fundamental questions remain unanswered. But the tools we now possess are far more powerful than ever before. The synergy between AI-driven computation and experimental validation holds the greatest promise for breakthroughs. Neither approach alone is sufficient. Understanding life's origin requires integrating insights from chemistry, biology, physics, mathematics, computing, geology, and even philosophy. Artificial intelligence provides the computational power to synthesize these diverse perspectives. For the first time in human history, we have tools powerful enough that the fundamental question of how life began may be answered within our lifetime. This is one of the most exciting frontiers in all of science. Thank you for your attention. The intersection of artificial intelligence and origin of life research represents one of the most profound scientific endeavors of our time, combining cutting-edge technology with humanity's deepest questions about our place in the universe.